our lecture number 7 on bulk soluble and surface active nanomaterials for biosurfaces by our foreign expert Dr. Mandeep Singh Bakshi. Thank you.
Good morning, everybody. We are starting with the next lecture. So here we will talk about the fundamentals of bioapplicability. We will try to understand that which important components of epithelial molecules are responsible to initiate the bioactivity, and then slowly we will move towards the surface active magnetic nanoparticles. You have already seen this picture. So the bioactivity which I am going to explain, so I'll be taking the example of respiratory system, how we can understand the bioactivity in terms of nanometallic pollutions and how we can prevent that. So these are the few things we will try to understand. This one you have already seen, so this interface is very important. This is lined with the surface active molecules between the lung air phase, that is the air spaces, in alveoli, and then the fluid phase. And this is the one that is disrupted bilayer of the interfacial surface active molecules. So it could happen because of any reason, but the main purpose here we will be talking about that what causes it. Today we are going to talk about that. So that is the suspended nanoparticles in the form of bioactive ingredients which have the ability to instantaneously interact with such kind of bilayers. Last time we were told that the pulmonary surfactants which help us in the breathing processes, so they are mainly made up of lipid molecules, DPPC 70% and POPG 30%. So these are the molecular structure. You could see that they are the zwitterionic. This one is particularly the zwitterionic molecule with double hydrocarbon chain, and in this case POPG is the sodium salt, and it is the anionic molecule because there is a negative charge on the phosphate, and there is one unsaturation in the hydrocarbon chain. So the combination of all these lipid molecules, they create the bilayer assembly that is adsorbed at the air alveolus aqueous interface. Apart from that, there are surfactant proteins. So there are 4. 2 are hydrophilic, 2 are hydrophobic. If you are the student of biology or at some point you have studied biology, you know what is the meaning of a protein. So proteins vary from one another because of the sequence of the amino acid. So all proteins are predominantly made up of amino acids. Only the sequence of the amino acids, so they are going to configure the properties of the protein. Some of the proteins are water soluble, some of the proteins are water insoluble. When the number of amino acids which are water soluble, they exceeds that exceeds the number of water insoluble or predominantly non-polar amino acids. So then the protein is water soluble, otherwise it is water insoluble. So water soluble proteins are known as hydrophilic proteins. The water insoluble or sparingly soluble are predominantly hydrophobic proteins. Protein, it uh, is not excluding the 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 possibility of hydrophobic domains in the case of hydrophilic and situation for including the possibility of hydrophilic domains. Schematically, I can say that this is a protein molecule. Okay, so you may have somewhere over here predominantly hydrophilic and hydrophobic. If I say this is hydrophilic uh, domain and this one is hydrophobic, or sometime the hydrophobic domains are hidden inside the protein aggregate structure by the hydrophilic domains. So when the protein unfolds, something it is like this, and sometimes you will see that the hydrophobic domain, if this is the hydrophobic domain, it comes in the vicinity of aqueous phase. So this is known as protein folding. There's a native structure of the protein. Native structure exists at a particular temperature and the pH. And when we alter the environmental conditions, we can unfold the protein. So that is the meaning of the hydrophilic and the hydrophobic. What we have done in our lab, we created the artificial lung in the apparatus. Now, it is not exactly the way we have the lung in the human body. It is a kind of apparatus where the functioning of the apparatus depicts the functioning of the lung. In the lung, you have so many alveoli. So here we have just picked a bubble and we depict this is a one alveolus. Now this is one alveolus. It is exactly at the interface of air and water. Now the surface lining of this air bubble, that is lined with the same composition of the pulmonary surfactants which exist in the human lung. So the same composition, the composition of DPPC 70% POPG, we mix these components along with the surfactant proteins and we inject it into the bubble. When we injected into the bubble, obviously they created the bilayer the way it is there over here, this bilayer. Now, we know that when we compress the lung, the surface activity should go to minimum value. Only then we can completely expand, expel the air. So what we did mechanically, the same amount of the pressure which the human body is exerting on the lung, we perform the same kind of experiment by adding the pressure over this. That squeeze the bubble and simultaneously we measure the surface tension. So what we are going to do, we are adding the compression. You could see the relative area. Adding the compression and we measuring the surface tension. Initially when we have not applied any pressure, or you can say that we have already air in the lung and the lung is filled with the air, and then we are applying the pressure to expel the air. So the amount of pressure which the human body is exerting on the lungs, that is very, very tiny. So that amount of pressure we are going to apply it over here. And when we apply that, we could measure the surface tension exactly in the same way as is happening in the human lung. So the surface tension reduces to close to zero value. You see, it is initially slightly above than the 30 millinewton per meter. And when we compress it up to over here, and you could see that the surface tension reduces to a minimum value. It means that the surface tension of the bubble, when it reduces to minimum value, the bubble compresses. The bubble compresses, and whatsoever is there that is completely expelled out, right? So the rate with which the human lung expands and compresses, the same rate we applied, and we try to measure the reproducibility of the surface activity. So you could see that the red plot, the color, the points in the red color, they are representing the compression when we put the pressure on the lung, and the blue, so they represent the expansion. And this clearly shows that the combination which is created by the amphiphilic molecules along with the surfactant protein in the human lung, the way that is working under the natural circumstances in the case of every human being, same composition is working exactly under the experimental calendar. Zero, practically there is no surface tension. If there is no surface tension, the bubble will not be like this, but it will be more compressed. Means this is somewhat close to the compression, but otherwise the bubble will be more expanded under the normal circumstances. What is the meaning of this? The meaning of this is that for the healthy lung, for a normal person, so you have something like this that is being performed by every alveolus. You have thousands and thousands of alveolus in the lung. So here this is just one. So it means that every alveolus in the human lung, so that is performing exactly the same kind of expansion and compression you could see in this experiment. And the value for the surface activity or the surface tension when the air is expelled, exactly that is close to zero under the natural circumstances, and under experimental circumstances, exactly that is also close to the natural value. It means the composition which we are predicting, which we have 70%, 30%, and it is close to 2% usually, the surfactant protein. It means that in the human lung, almost close to exactly this composition exists. Now, this is the story for the normal circumstances when there is no pollution. We are going to consider the pollution in the form of nanoparticle. You could imagine what if we have pollutants here, dust particle in the form of nanoparticle, and if you are going to inhale them, what is going to happen? So, a pollution model, synthesis of gold nanoparticle capped with primary surfactant as pollutants. In vitro use of gold nanoparticles as pollutant to study their effect on the breathing process. What we did, in order to estimate the effect of the pollutant on the respiratory system, rather than picking the dust particle from the air as the sample, what we did, we picked the gold nanoparticle. The reason was that we synthesized the gold nanoparticle of same shape and size, and then we use them over here to just know that if I'm going to inject the gold nanoparticle into the alveolus, it is exactly the same way as if you are breathing the nanoparticle and they are going into your lung and they are residing in the alveoli. So then what will happen? what we did, we synthesized the gold nanoparticle the way I have already told you, and the stabilization of the gold nanoparticle I have already told you that how we can provide the bilayer. And here, the bilayer of the gold nanoparticle is provided with the phospholipid molecules, exactly those which I showed you. And we also saw the bilayer in the transmission electron microscope. So what we did actually, what is the meaning of this? We initially prepared the gold nanoparticle without the presence of the lipid, exactly those lipid molecules which we have in our lung at the same composition. And in that solution, we added our gold nanoparticle, which was synthesized the way you did yesterday in the lab. We purified them. We just added them in combination of the polymerase surfactant suspension, and then we tried to disperse them, and we saw after the mixing, and we get the TEM image, and we saw that the polymerase surfactant, they created the bilayer around each gold nanoparticle. The most strange results were when we added the surfactant protein, SPB. This represents the surfactant protein which I am mentioning over here. SPA, SPB, SPC, and SPB. We pick the SPB, it is much more surface active. When we added SPB, the gold nanoparticle, which were less segregated in the presence of lipid molecules of the same composition as of human lung, as we added tiny amount of the surfactant protein, the aggregation was enhanced. You see the aggregation? There was less aggregation over here, but as soon as we added tiny amount of the surfactant protein suspension, and then we got the image, and we saw that the aggregation of the gold nanoparticle is now much more enhanced than what is over there. What is the meaning of this? I'm just telling you now. I'll explain what is the meaning of this. It means that the lung surfactants which we have in the human body, they have very high affinity for a nanoparticle. Nanoparticle could be from the gold, could be from the silver, or that could be a dust nanoparticle. They all have very high affinity for the polymerase surfactant complexation. As soon as the particle comes in contact with the bilayer in the human lung, that bilayer instantaneously codes that nanoparticle. No matter that nanoparticle is made up of which kind of substance, whether it could be a transition metal or any kind of oxide like silicon oxide minerals of dust. How do we know this? How do we know that this is exactly happening when you inhale the pollutant particles in your lung? I'm now taking you to that part. So you see that this was the one image, and this is the next one, where before adding SPB you have an image like this, but as soon as you added SPB, surfactant protein B, you see a very intense aggregation. You see the gold nanoparticle? Read the scale bar. The gold nanoparticle is less than 10 nanometer and very clear spherical gold nanoparticle, right? Okay. Now, as I told you that in order to become a good scientist, you have to look for a technique which can easily apply for your easy detection or the step in which you have to go ahead. I mean to say that suppose if you want to see that there is an aggregation here or there is aggregation here, then what is the easiest technique? The transmission electron microscopy which I am showing over here, probably it is not available everywhere and it's not that easy to instantaneously get the image or the sample. But what is much easier to perform? That is the UV visible spectroscopy. So everywhere you have the visible spectroscopy. What you could do, you could take the gold nanoparticle, the solid red line with POPG, gold nanoparticle coated with POPG, making the bilayer. Then the gold nanoparticle coated with DMPG. I have already explained the nomenclature for these molecules in the previous slides. So when we take these samples without the surfactant protein, you see the sharp band here. That is because of the surface plasma resonance of the gold nanoparticle or the absorbance which you measured yesterday. You prepared the gold nanoparticle suspension and you took the absorbance, and there was a peak close to 500 nanometer. That is the one over here you see close to 520 nanometer. In the same sample, when we added a tiny amount of surfactant protein B, the same kind of suspension of the surfactant protein B, this peak instantaneously shifts to at a higher wavelength with much broader peak. The intensity is much higher over here in comparison to this. So, greater the intensity, greater is the number of nanoparticles. What is the meaning of the shifting towards the higher wavelength? Whenever you have the aggregation in the nanoparticle from here to here, any aggregation which happens in the gold nanoparticle suspension, it instantaneously shifts the gold nanoparticle absorbance to the higher wavelength. So that's what's happening. Apart from that, that peak must be much broader in comparison to the peak that was without the aggregation. So this is without the aggregation, or the tiny amount of the aggregation of the gold nanoparticle. This clearly shows that there is an intense aggregation of the gold nanoparticle. How it happened? This is the first evidence. This is the simplest way of determining that what is happening in the solution. Thereafter, you can perform the microscopic imaging studies slowly, but at least you know that by adding surfactant protein, aggregation is taking place. Now, another important thing, the color of the gold nanoparticle suspension, if the absorbance is close to 520 nanometer, it should be ruby red color. But you are not expecting the ruby red color close to 700 nanometer. It's a visible region, right? So in the visible region, as you are moving toward the towards the higher wavelength, the color changes from ruby red to purple and some other kind of shaded between ruby red and the purple. Now, it shows that the aggregation has happened. You can perform another very simple experiment, that is the gel electrophoresis. It is everywhere in the biology lab. What you could do, the same sample without the surfactant protein B, if you are going to put it in the gel well here for POPG, apply the voltage, you see there is a transfer of the electrons, the transfer of the nanoparticles towards the positively charged electrons, because these POPG are predominantly negatively charged. So hence the shell that is being created, the bilayer shell that is being created on the surface of the nanoparticle, that is predominantly negatively charged. So it means that they are going to move towards the positively charged electron. But as soon as in the same sample, if you are going to add the surfactant protein B, now the aggregates are very large enough, they cannot move through the channel of the gel. So hence there is no shifting of the aggregated gold nanoparticle in the presence of surfactant protein B and surfactant protein B over here. This is another evidence that intense aggregation has taken place. But you will be wondering how it is related to respiration. I'm coming to that point. What is happening schematically? This is one gold nanoparticle, this is another gold nanoparticle, this is another one. This is the bilayer of the polymerase surfactant. BMPG or POPG, you can say, bilayer of the phospholipid molecule. Exactly those molecules which are there in the lung. Now, initially they are separated from one another. They have the colloidal stabilization in the solution, fine regular suspension, colloidal suspension. But as soon as I'm going to add the surfactant protein B, this is the overall schematic representation of surfactant protein B. So it is showing the various domains. You don't need to go through that. When we add this, the surfactant protein B, which I have shown schematically over here, that has the ability to bind with the bilayer of the adjoining nanoparticle. So therefore, it is acting as a glue. It is facilitating the aggregation in this way. Different nanoparticles which were quite separately separated from one another, suspended in the aqueous solution in the absence of surfactant protein B. But as soon as we are going to add surfactant protein B, this kind of association, surfactant protein B with the polymerase surfactant, instantaneously induce the aggregation, and that aggregation shifts the absorbance to the higher wavelength and stops the displacement of the aggregates through the gel network. Okay, so now we are coming back to our artificial lung. So this was the normal artificial lung functioning without the pollutants. Now we have created the model pollutants, gold nanoparticle with phospholipid. They act as a model, not exactly the pollutants which you have in the air, but they can work exactly or very closely similar to that of the pollutants. What we did, we picked the sample of the gold nanoparticle. This is the sample, the gold nanoparticle, and we added DPPC, POPG and SPB in the gold we took this suspension actually, and we added the purified gold nanoparticle, and we produce a stable suspension. Now this suspension we injected into your artificial lung or artificial alveolus. This is artificial alveolus. And we performed the same experiment. Same experiment, expecting that after the compression, the surface tension should reduce close to zero as it was happening earlier. See over here. It was coming close to zero in the absence of pollutant. But when we added the gold nanoparticle as pollutant, as model pollutant, so we were expecting that the surface tension after the compression should come in close to zero, but that is not happening. So the surface, you see, initially that could be higher value, but it is the minimum surface tension we are achieving close to only 20. This value should have gone to touch 0 millinewton per meter, but that is not happening. What is the meaning for this? It means that the compression with the pressure we apply to expel the air or to squeeze the bubble, now that is not working. Why it is not working? Because the bilayer, the interfacial bilayer that is present at the air and the aqueous interface, that is not possessing the sufficient amount of the surface activity that could reduce the surface tension. What is the meaning for this? It means that if the pollutants are going into the human lung, then the functioning of the human lung is impeded exactly the same way as is happening over here in the experimental artificial lung. Okay, so this is a clear evidence. It means that when the pollutants are added, this is the natural surfactant. Without, if I do not add the pollutants, there is no problem. The artificial lung experiment is working exactly the natural lung. But when I add the pollutants, then the artificial lung is not working as a natural lung. So it means that the artificial experiment clearly shows that the pollutants have impeding effect on our respiratory system. Now we will try to look into the microscopic picture of the bilayer in the lung. What is happening? I told you that the bilayer is created by the vesicular system. It is the vesicle. I have shown just part of the vesicle over here. Reminding you once again which were the vesicle we were talking about. These were the vesicles. Lamellar bodies. They were going over here, opening up, producing the bilayer. Right? Over there you can see opening the bilayer. So I'm talking about it. And I'm just taking the part of the sketch to explain microscopically what is happening at the molecular level. So, this is air-liquid interface. You see the vesicle that is coming at the interface and then opens up and creates the bilayer. Okay? So, in the bilayer, this bilayer is created. It has, I have shown only the monolayer, but because the reason is air is non-polar, so therefore the upper side should be lined with the hydrocarbon tails, hydrophobic tails, rather than the head group. So head groups are inserted only in the lung fluid, that is the aqueous phase at the interface. So that is why you see only the monolayer exactly at the interface. Proposed mechanism for the pollutants absorption at the air alveolar interface. Injured lung means impeding the functioning of the lung. How that is working? Okay, these are the gold nanoparticles. These are the gold nanoparticles. All the pollutants, they are coming from where? They are coming from the air, not from this side. There is no way the nanoparticle travel through the body and enter, and they can come at close to the lung. So what you inhale the air, if that is polluted with the pollutants, so then the nanoparticles are going to hit the layer, interfacial layer of the phospholipid molecules. As soon as they are going to hit it, instantaneously the phospholipid molecules will create a bilayer. This is the bilayer. You have proved this because you have proved this when you are adding the gold nanoparticle in the phospholipid suspension, you are creating a bilayer. So you have already seen in the experiment, and that is what is happening under natural circumstances when you face the pollutant. So now, so what if the pollutants are, because everybody is inhaling dust particle and everybody is very healthy, right? So there is no issue. Yes, absolutely there is no issue. Where they have gone? They are taken up by any kind of foreign particle, living or non-living, that enters into the lung during the respiration is taken up by macrophages, alveolar macrophages. They are there in the lung fluid. So any foreign particle that enters through the interfacial layer instantaneously taken up by the macrophages. Suppose if you ask me, so we know that for the soft particle or any kind of living organism that enters into the lung during the respiration, right? So we can understand that these macrophages, they have the ability to kill it and to split it. But the problem is these particles are the metallic particle, crystalline particle. Our human body has no ability to disintegrate the crystal into its component. That is not going to happen. So then, when you inhale the dust particle, now those are the silicon oxide minerals. Where they are going, right? So this, I think the better answer probably professor of medicine can give you. I don't have that answer that where they end up eventually, but that is a very, very interesting part in biology. But the thing is, why would the respiratory problems are going to arise? Because of the pollution. It means that something is happening. Something is happening. Okay, so I have 15 minutes. Sorry. Okay, something is happening. Something is happening with this mechanism. The rate with which the particles are entering into the lung and the rate with which they are being cleared by the alveolar macrophages, if that is going to be disrupted, then the problem arises, right? As long as the rate is the same, there is no issue. Every day you are inhaling those particles. So much pollution is there. But those people where this clearance from the lung fluid by the alveolar macrophages is going to be disturbed, only then there's a problem. Then, what is the problem then? Then the particles will remain here. The concentration of the particle is high rather than the rate with which they are being cleared from the alveolar macrophages, and hence the particle will start residing over there and will disrupt the continuity of the interfacial surfactant layer which provides the surface activity. The one I have shown over here. In this diagram I show, but this could be because of your other health issue, biological health issue, but if we have to look at the pollutants, it will become a permanent issue because you have a certain concentration of the pollutant. It is always there because you are constantly exposed to the pollutant. And the alveolar macrophages, those are like this. They have not that much ability or capacity to remove all the pollutants. It means that your interfacial phospholipid layer along with the surfactant protein can never be a continuous film, and hence you cannot have the close to zero surface tension during the respiratory expansion or compression. This is the thing which shows over here. Okay, so this continuous film is required in order to perform the healthy respiration. When it is disrupted, so these particles are already there. Macrophages has limited ability to clear them, and now you see it's a non-continuous, so therefore it cannot provide the required amount of surface activity. So now it means that this film is now uncompressed. It cannot be compressed to the value that leads to the surface activity close to 0 millinewton per meter, right? Since this part is not lined with that of the phospholipid molecules like this part, so it's a non-continuous part. So now, on the basis of this, we can conclude that metal nanoparticle as air pollutants have strong interaction with pulmonary surfactant. This is the metal nanoparticle as model. But if you ask me, what if there are dust particle or the carbon suspended fiber or the particle from the fossil emission? They are also having the same kind of entity. Maybe there could be a slight variation because this is the surface of the transition metal as a gold nanoparticle for the silicon oxide. So they are the silicon oxide mineral particles. surface properties are not exactly the same as that of the transition metal nanoparticle. And for the carbon particles, they are completely non-polar. Obviously, their surface properties are much different. So in all the cases, but the phospholipid molecules, so they have the dual affinity to interact with the polar as well as the non-polar surfaces. So that no matter the particle which you are going to inhale, they are polar, they are possessing the charged surfaces or the non-polar surfaces. They are going to be complex with that of the phospholipid molecules because the phospholipid molecules have dual ability to interact with both the charged surfaces as well as the non-charged surfaces. Constant exposure of air pollutants can produce respiratory disorder, and this is the problem which we see normally happening whenever we have a constant rate of high pollutant, concentration of the high pollutants suspended in the air. So it means that you cannot avoid the respiratory problem, and particularly the elderly people, they are more prone for that because of the less resistance, and hence the alveolar macrophages, they are not that active to instantaneously remove the pollutants as much as in the case of the young people. But a very intense amount of the suspended particle pollution, so that is going to affect everybody, no matter in which range of you lie in your life, for the children, adults, or the elderly. So just concluding statement. So we started our lecture by talking about the bulk soluble and the surface active nanomaterials. You see, the nanomaterials we have shown in this lecture, there is no doubt they are bulk soluble because they are coated with the phospholipid layer. But when they interact with the surfactant protein, they absorb at the biointerfaces. The lung surface is the biointerface. There is air phase as well as the lung fluid phase, that is the aqueous phase. So therefore, we know the impeding effect of, or the intense toxic effect of the pollutant, how they can interact with the biointerfaces. So this is just one example. This is not the only example how the pollutants are going to interact with the biointerfaces. We have so many biointerfaces. In our alimentary canal, we have also biointerfaces. Everywhere on the open wound, we have the biointerfaces. So it means that we have plenty of other examples. Suppose if you want to read more article or more information to gain in this direction, I will say that just look for the publications which talk about the interaction of the pollutant with the biosurfaces, and you will have much more information. Thank you so much for